Dear students, Assalamu alaikum. Today we are going to talk about our course Physiology and Immunology, course code CHEM444. This is lecture number 4 of our series of online lectures. The topic of our today's lecture is Biochemistry of Sweet. In the outline of today's lecture is we will talk about what is sweet. Then we will talk about types of sweet glands. Then we will talk about function of sweet. Then we will talk about characteristics of sweet. This will be followed by talking about factors affecting the composition of sweet, and then we will briefly talk about sweeting abnormalities. Let's go to talk about what is sweet. It is a clear salty liquid secreted from the skin by the sweet glands. So sweet is produced by sweet glands and it is a clear salty liquid. Sweet glands are small tubular glands situated within and under the skin. The average person has 2.6 million sweet glands in their skin. Sweet glands are distributed over the entire body except for certain parts of the body like lips, nipples and external genital organs. This is a diagram showing skin anatomy. Here in this skin anatomy diagram we see different parts of the skin. This is stratum corneum, new skin layer, then subcutaneous layer. You can see here fat cells. One can see sebaceous gland. Here are the hair follicle. This is showing erector muscle, and here is the sweet gland. You can see here the sweet is poured in here and it is secreted through this sweet gland. This is the two parts of the skin. One is the epidermis and the other one is the dermis. So this diagram gives us an overall idea of skin anatomy, and it is showing the location of the sweet gland. Now we talk about types of sweet glands. There are broadly two types. One is called eccrine, the most numerous type that are found all over the body, particularly on the palms of the hands, soles of the feet, forehead and armpits. Then there is another type that is called apocrine. They are mostly confined to the armpits, axilla and the anal genital area. They typically end in hair follicles rather than pores. Now this diagram is showing the sweet pore. You can see it here. Here this is showing the hair follicle. This is the eccrine sweet gland. This is showing apocrine sweet gland. You can see it here. So this diagram overall summarizes the location of the two broad categories, eccrine sweet gland and apocrine sweet gland. So this gives you an idea of how these glands are localized. Now we talk about eccrine glands. they have smaller diameter. It ranges from 50 to 100 micrometer. These eccrine glands, they are active from birth. Apocrine glands become active only at puberty, produce a sweet that is free of proteins and fatty acids. Now, what is the amount of sweet produced depends upon our states of emotion and physical activity. Because you can see in your life, you would have observed that when the weather is cold, or if we are not physically active, we produce a less amount of sweet, whereas in situation like exercise or when the weather is hot, we produce a lot of sweet. So the amount can vary depending on situation. In case of narrow stimulation, we produce sweet, or in case of exercise, you can see the athlete producing sweet because of exercise. So one of the main stimulus that produces a lot of sweet. And of course, weather is very important. In hot weather, if we are doing some physical activity, or even if we are not doing physical activity, but we are not doing a lot of vigorous physical activity, but the weather is hot and we are just walking, we do produce a lot of sweet. And if the weather is cold, normally the production of sweet is low or limited. So the overall, it summarizes that the amount of sweet produced depends upon our states of emotion and physical activity. Higher the physical activity and hotter is the weather, more sweet is produced. Now, why, and the question comes to mind, why we produce sweet? What is the function of sweet? The most important function is thermoregulation, which is the principal function of sweet. With the help of sweeting allows the body to rid itself of excessive heat, producing excessive heat production through the evaporation of water, which brings about cooling of the body. Did you know that evaporation produces cooling effect? So this is why sweet production is so important, because when the outside temperature is hot, our body produces sweet and it evaporates from the body, and thus in this way it produces cooling effect, and it helps to maintain our body temperature at a certain level, like 37 degrees C, which is the normal body temperature. So in case the temperature, external temperature is high and our body wants to maintain its normal temperature, so it will produce sweet, and this will help in the thermoregulation of the body. So this is very important. Now we talk about characteristics of sweet. So normally sweet has no odor, but you see that if a person sweat for a longer period of time, one can sense some smell. This comes from reaction with bacteria on the skin and can produce an unpleasant smell. So basically, the unpleasant smell that can be seen after sweating is the result of reaction with bacteria on the skin. Otherwise, normally sweat has no odor. Maximum volumes of sweet is around 1 liter per hour in a hot climate. This is approximately the amount of sweet that is produced per hour in a hot climate. Now we talk about the composition of the primary secretion. It is very similar to that of plasma, except that it does not contain plasma protein. The specific gravity of sweet ranges from 1.002 to 1.005. The pH of sweet is from 4.5 to 7.5. Sodium ion that is present in sweet is 30 to 70 milligram. Potassium ion is 0 to 5 milligram. Chloride ion is 30 to 70 milligram or less. Urea is 4 to 6 times higher than in plasma. And lactic acid is higher than in plasma, especially in athletes. It's because athletes, when they apply a lot of force, they do pass through some type of anaerobic respiration, which results in higher amount of lactic acid production. So this is why it is higher than in plasma, especially in athletes. In normal circumstances, we may not produce a lot of lactic acid, but in case of athletes, they do pass through higher level of anaerobic respiration, resulting in production of greater amount of lactic acid, which is then reflected in the sweet in higher amount. Then glucose can be found in a very small amount. So this slide gives us an overall picture of how the composition of sweet look like. So there are a lot of ions, urea and lactic acid in case of athletes, and this is the pH. So this slide summarizes overall the composition of sweet. Now there are certain factors affecting the composition of sweet. These factors include when we have low sweet rate, that is we are in a resting position or in cool temperature, the primary fluid passes through the duct very slowly. Sodium ion and chloride ion are reabsorbed, which reduces the osmotic pressure of the fluid, leading to water reabsorption and concentrating most of the other constituents. Thus, at low sweeting rates, the sweet produced is low in volume and is more concentrated in constituents such as urea, lactic acid and potassium ion, and less concentrated in sodium ion and chloride ion. Now at high sweet rate, that is when we are either working or we are doing exercise, or if the temperature, external temperature is hot. So in this situation, when the sweet glands are strongly stimulated, large amount of primary secretion are formed which will flow too rapidly through the sweet duct, reducing the chance of sodium ion and chloride ion reabsorption. Thus, water reabsorption is also reduced, leading eventually to the production of a large volume and less concentrated sweet. So this is the situation when the production of sweet is high, at higher rate in case of work, in case of exercise, or in case of hot temperature. This is the diagram showing in case of low flow and in case of higher flow. So you see the sodium ion, chloride ion in water in lesser amount. So in low flow, see sodium ion, chloride ion in water. This is high. This is the dermis region. In the interstitial fluids are high. It's sodium ion, chloride ion, and low potassium ion. Here is water, sodium ion, chloride ion, and potassium ion, and showing the movement of ions in water in making sweet. So these are the different constituents which are showing the movement of ions in water in making sweet. Now there are certain abnormalities associated with the process of sweeting. The one of those abnormalities is hyperhidrosis. Hyperhidrosis means it is abnormally increased sweating. It means that when the sweating is higher than normal. So there are different reasons that can cause this sweating abnormality or hyperhidrosis. One of these abnormalities is obesity, that is high weight. Obesity is one of the reasons of hyperhidrosis. Then there is another reason that is associated with sweating abnormalities is hyperthyroidism. Hyperthyroidism results in hyperhidrosis or increased sweat production. Diabetes is another cause that is associated with hyperhidrosis process, and there are certain medications that can be responsible for producing hyperhidrosis, because metabolism of certain diseases, certain medications, they can result in production of heat, which can result in process of sweating in order to counter the heat production, because we know that sweating does produce cooling effect. So this is hyperhidrosis that can be caused by some of these diseases. Now there is another types of sweating abnormalities that is called hypohidrosis. It is a partial loss of sweating, while there is another extreme situation which is called anhidrosis. It is a complete lack of sweating. This can occur for a number of reasons, which include some skin disorders, burns to skin that damage the sweat glands, underactive thyroid or hypothyroidism, and a situation called dehydration, because if the amount of water inside the body will be low, it will result in hypohidrosis situation, because there will be not a lot of water for the body to excrete it, and the body will try to retain most of the water for its needs. So this situation can result in hypohidrosis. Now there is a sweet taste. It's called chloride sweet taste. What it does is that it measures the concentration of chloride that is excreted in sweet and is used to screen for cystic fibrosis. In cystic fibrosis, transmembrane conductance regulator or CFTR chloride channel is defective, thus preventing chloride to be reabsorbed into sweet duct cells. Consequently, more chloride stays in the duct, leading to an elevated concentrations of chloride in sweet of individuals with cystic fibrosis. So this sweet taste can be helpful in understanding cystic fibrosis, and this is performed. This chloride sweet test is performed to get to know about the disease which is caused by defective chloride channel. This is all about sweet. I hope you enjoyed the lecture, and if you have any question, I will be very happy to answer that. Thank you very much for your attention. And see you in the next class.